All right, guys, let's work on some biological psychology. First thing we're going to cover is learning and memory. Learning and memory. So, with learning and memory, you have habituation and sensitization. Habituation and sensitization. Learning and memory. Habituation and sensitization. And the differences and what they are. So, type of learning. Habituation. Habituation is a response to a stimulus, gradually gets smaller over time. So a decrease of response. So habituation, response to a stimulus gradually gets smaller over time. It becomes a habit. You form a habit. You become habituated to something. So the response to a stimulus gradually gets smaller over time. If you turn on the fan in a room, you will initially hear it. Eventually, you don't hear it anymore. That's an example. Turning on a fan in the room, or maybe some sort of air conditioning noise in a room. You don't hear it. At first you do, and then you become habituated to it. So response to a stimulus gets smaller gradually over time. Decrease of response. If you turn on a fan in a room, you initially hear it. Eventually you don't hear it anymore. So that's a habituation. Sensitization is the opposite of habituation. Response to a stimulus increases over time. After a bumpy plane ride, you may be a little jumpy for a while. That's sensitization. Or let's say you get spooked about something. Something kind of gets you a little alert. Or after a traumatic situation, you almost get into a car accident and then somebody says, hey, you okay? You get all nervous about it, right? So you become sensitive to it. It's a sensitization. Response to a stimulus increases over time. And then habituation was a response to a stimulus gradually decreases or gets smaller over time. The response, decrease of response to a stimulus. Neural mechanisms of learning. So, neural mechanisms of learning, largely studied in Aplysia californica, which is an animal. So they studied on an animal called an Aplysia californica. They have larger neurons and fewer neurons, make it a good animal to study. So it's sort of an ocean creature. They have gills and a siphon. So the gill withdrawal reflex. The gill withdrawal reflex. So the gill is used for breathing. The gill is covered with mantle shelf. The gill is for breathing. The gill is covered with the mantle shelf. Waste and seawater are released through the siphon. Touching the siphon leads to a retraction of the gill. So if you touch the siphon, the gill retracts, right? So the gill withdrawal reflex. The gill is used for breathing. The gill is covered with the mantle shelf. Waste and seawater are released through the siphon. Touching the siphon leads to a retraction of the gill. So how does that have to do with rehabilitation, or not rehabilitation, habituation? There's three hypotheses. Touching the siphon repeatedly leads to less of a reflex. So the animal, the Aplysia californica, if you keep touching the siphon repeatedly, leads to less of a reflex or retraction of the gill. Why would this be? There's three hypotheses. Number one, either the sensory neurons are becoming less responsive. The neurons at the siphon, either the sensory neurons at the siphon are becoming less responsive. Number two, motor neurons are becoming fatigued. So sensory neurons are becoming less responsive. Maybe the motor neurons are becoming tired. Remember, these are hypotheses. They're just sort of, from the information they gathered, they're thinking this might be it, right? The empirical data. So the sensory neurons are becoming less responsive. Maybe this animal, the motor neurons, and with this animal are just tired, or changes occur between both the sensory and the motor neurons. So these are the hypotheses. So what happens is they touch the siphon repeatedly, and that leads to less of a reflex or less of a retraction of the gill. And they're like, why is this happening? It could either be the sensory neurons are becoming less responsive, the motor neurons are becoming tired, fatigued, or changes occur between both the sensory and the motor neurons. So what is it? Habituation explained. Single cell recordings show that sensory neurons do not decrease activity with each touch of the siphon. So it's not that they're becoming less responsive. So single cell recordings show that sensory neurons do not decrease activity with each touch of the siphon. Number 2, electrical stimulation of the gill motors neurons show that it can still contract after being habituated. So it's not becoming tired. So changes must be occurring between the sensory and the motor neurons. So it's the third hypothesis. Changes occur between the sensory and motor neurons. So repeated siphon touches lead to less calcium into the sensory neuron, which means less neurotransmitter is being released into the synapse. So like the body is not regenerating the calcium so much because repeated siphon touches lead to less calcium, Ca plus, into the sensory neuron, which means less neurotransmitter is being released into the synapse. Smaller EPSPs occur in the interneurons and motor neurons. So smaller excitatory postsynaptic potentials? I don't know what the last P means, but smaller EPSs occur. So smaller excitatory postsynaptic potentials occur in the interneurons and motor neurons, leads to smaller gill withdrawal. So again, it just sort of has to do with the less calcium, less of that action potential into the sensory neuron. Less Ca plus action potential, less of an electrical signal into the sensory neurons. It's like they're being used too much, right? They're being depleted. Causes less activity in gill. That's electrical signal. And so smaller EPSPs occur in the interneurons and motor neurons, lead to a smaller gill withdrawal. So it sort of is a motor thing. It's because it's the the action potentials occur, or the smaller action potentials, excitatory postsynaptic potentials occur in interneurons and motor neurons, lead to a smaller gill withdrawal. So sensitization. After an electrical shock to the head or tail, aplysia show an increased gill withdrawal response. So sensitization. So this is kind of what they're doing to try to figure if they become sensitive to this thing. So after an electrical shock to the head or tail, the Elypsia californica show an increased gill withdrawal, like trying to protect itself. Gill withdrawal response, an increased gill withdrawal response. Sensory neurons release more neurotransmitters onto the interneurons and motor neurons, which leads to larger response. So sensory neurons release more neurotransmitter. So long-term changes were observed as well. Normal aplysia showed 1,300 axon terminals synapsed with sensory neurons. So normal aplysia showed 1,300 axon terminals synapsed with sensory neurons. Those that experienced sensitization had 2,800 terminals. Those that experienced habituation had 800 terminals. So when you became, when the response to the stimulus decreased because of forming a habit, there was only 800 terminals now. 800 axon terminals synapsed with the sensory neurons. But when you created a sensitization, right, when you created this sensitization, when that response to a stimulus increased over time, the normal aplysia now had over 2,800 axon terminals synapsed with sensory neurons. So it became not just a chemical change, but a structural change. I mean, not just a chemical change, but a structural change. Remember the synapse, the axon terminals synapse, which became a. Remember the synapses are important when you create a habit with anything. That's why I'm reading a book on desires. That suppose they think we're not addicted to anything. We just have this. We create a new form. We form a new habit. Our synapses create new connections, which make this habit more stronger and meaningful, which would sort of create this sort of interwebbing of more difficult to get to change that habit. So long-term potentiation. So long-term potentiation. Learn more of what preferred. Unlearning of something you want to do. Phenomena, LTP. So long-term potentiation is a phenomena. It's also called LTP, long-term potentiation. So it's sort of this theoretical thing, right? So long-term potentiation, a long-lasting strengthening of a synapse. More neurotransmitter is released with any input, thought to be associated with memory formation. Long-term depression, a weakening of a synapse that lasts hours or days, thought to be associated with learning. So unlearning of something you want to not do, the unlearning of something you no longer want to do. Not that it's a negative, not that it's a depression, like, oh, like, that person has Alzheimer's. No. They think that a weakening of a synapse. So for your brain to unlearn something, right? It's a phenomena. It's LTP, long-term potentiation. So let's go to the first one, a long-lasting strengthening of a synapse. More neurotransmitter is released with any input. So more neurotransmitter is released with any input, supporting, learn more of what you prefer or what's preferred. So learning more of something that you actually prefer to learn, not to be associated with memory formation. Long-term depression, a weakening of a synapse that lasts hours or days, thought to be associated with learning, unlearning of something you want to not do. So long-term depression, long-term depression, and then also long-lasting strengthening of a synapse. So cellular mechanism for LTP, long-term potentiation. NMDA and AMPA receptors. Both are receptors for glutamate. Remember, the main excitatory neurotransmitter. Normally, just NMDA receptors are available to depolarize the postsynaptic membrane. Normally, just NMDA receptors are available to depolarize the postsynaptic membrane. When the connection is strengthened, AMPA, which amplifies signal coming in, are added to the membrane. So AMPA supports the NMDA receptors, which are normally just the ones that are available to depolarize the postsynaptic membrane. Allows for the postsynaptic membrane to be depolarized easier. LTP, long-term potentiation. So is LTP the mechanism for long-term memory? Both can last indefinitely, NMPA and AMPA. No, wait. So I think what he's saying is the long-term potentiation the mechanism for long-term memory? Is it called the LTM? So when it says right here, both can last indefinitely, I think he's talking about both long-term potentiation and long-term memory. Both can result from very brief input. So you can have a brief scenario in life, a brief life experience, and have it remain in your brain for a long time. Remember, long-term potentiation is a long-lasting strengthening of a synapse associated with memory formation, but then it's learning more of what you prefer. Long-term potentiation is learning more of what you prefer. Long-term depression, unlearning of something you want to not do. A weakening of a synapse that lasts hours. So something that strengthens the synapse so you could learn something you prefer and then unlearn something you don't want to do is long-term depression. Weakening of the synapse not to be associated with learning. Because if you want to learn something, then maybe unlearn it, right? And then memory, you want to remember how to do something. So you want to remember it. Something you learn, you want to forget it, so you want to unlearn it. So both can result from very brief input. LTP is consistent with models proposed by Donald Hebb, who was a mathematician. Long-term potentiation is found in cells thought to be associated with memory formation, like the hippocampus and the cerebellum. So long-term potentiation is found in cells thought to be associated with memory formation, like the hippocampus and the cerebellum. NMDA receptors that have been linked to memory are also linked to LTP. What about the brain structures? What about the brain structures? Learning and memory is the objective. And so, what about the brain structures? We're worried about the hippocampus and the temporal lobe. So the hippocampus is part of the temporal lobe. And also the thalamus. The Papez's circuit, the prefrontal cortex, the basal ganglia, and the cerebellum. So learning and memory is the objective. What about the brain structures? The hippocampus and the temporal lobe, the thalamus, Papez's circuit, prefrontal cortex, basal ganglia, and the cerebellum. Learning about patient HM. Large areas of HM's hippocampi and temporal lobes were surgically removed. The hippocampi and the temporal lobes were surgically removed. This has to do with this patient. We're discussing a patient. Personality and IQ were not impacted. Experienced profound anterograde amnesia. We're gonna learn what that is. So experience profound. So personality and IQ were not affected. Large area of patient HM's hippocampi and temporal lobes were surgically removed. Personality and IQ were not impacted. Experienced profound anterograde amnesia. So we're going to talk about amnesias. Retrograde. Retrograde has to do with past memories. Loss of memories of the past. Loss of memories of the past. Anterograde. Inability to form new memories. So, patient HM unable to form new memories, which is anterograde. Remember, retrograde is the inability or the loss of memories of the past, losing memories of the past. Anterograde is the inability to form new memories. Well, HM actually had issues with both, but had profound anterograde amnesia, but had issues with both. Anterograde and retrograde. More about HM. His short-term memory was okay, which is also known as his working memory. Short-term memory, aka working memory. He could learn procedural tasks. He could mirror drawing. His deficits were in explicit memory tasks. So he could learn procedural tasks like riding a bike, but explicit memory tasks like remembering facts, events, etc. So his short-term memory, his working memory was fine. He could learn procedural tasks like mirror drawings. His deficits were in explicit memory tasks, facts and events, etc. He had problems with those explicit memory tasks, but procedural tasks like riding a bike, he was fine, as long as he was taught or learned. So HM conclusions, patient HM conclusions. The ability to store memories and the ability to access previously stored memories are in different brain areas. So the ability to store memories and the ability to access previously stored memories are in different brain areas. Procedural memories are dealt with in different brain areas than explicit memories. So procedural memories are dealt with in different brain areas than explicit memories. Different parts of the hippocampus. The right hippocampus, excuse me, the right hippocampus is active during spatial memory. Remember, navigation, maps, where things are, like remembering where things are. So navigation and maps, remembering where things are. So the right hippocampus is active during spatial memory processing. The right hippocampus is active during spatial memory processing. So when you're navigating the world and remembering where things are, may include a three-dimensional representation of the world around. The left hippocampus is more active during verbal memory tasks. They think that there might be gender differences here. I don't know. I just have a little note here. I remember exactly why I put that. I don't know if it has to do with the hippocampus though. So different parts of the hippocampus. The right is active during spatial memory processing. May include a three-dimensional representation of the world map. The left hippocampus is more active during verbal memory tasks. So maybe explicit things, like not necessarily procedural. Explicit memory tasks. All right, Korsakoff syndrome. Another. We're done with the HM thing, right? So Korsakoff syndrome is caused by alcoholism. So it's caused by a thiamine deficiency due to alcoholism. A thiamine deficiency. Damage to the thalamus and the mammillary bodies. So this Korsakoff syndrome is caused by, or is due to damage to the thalamus and the mammillary bodies. It's a thiamine deficiency due to alcoholism, Korsakoff syndrome. So more with the thalamus. All right, patient NA. This is a different issue. Patient MA or NA, like not available. So patient NA. This is something that they discovered because of patient NA about the thalamus and how it works with memory and learning, right? What happened was it was a fencing foil produced a lesion to the left thalamus. So a little fencing sword, you know, a little when they wear a funny suit and they have like a little sword with the needle, and it's a sport. It's in the Olympics where you see it mostly. So a fencing foil produced a lesion to the left. I think it went like in his ear or something, or like his nose, and it poked his brain. So a fencing foil produced a lesion to the left thalamus. NA's amnesia was similar to HM's. Remember, patient HM had profound anterograde problems, but also had problems with retrograde. Anterograde was forming new memories, and also retrograde is the loss of old memories. So the DNM's task, delayed non-matching. So the DNMS or DNMS, the DNMS task, delayed non-matching to sample task. Monkeys with temporal damage or thalamus damage cannot do the DNMS task. So monkeys with the temporal damage, which is hippocampus damage or thalamus damage, cannot do the DNMS task. Requires the ability to transfer memories from short-term systems into the long-term systems. So with the hippocampus damage in the temporal lobe or thalamus damage, it affects the ability to transfer memories from short-term systems into long-term systems. All right, Papez's circuit, a network that connects the thalamus. So Papez's circuit, it is a network that connects the thalamus, mammillary bodies, and the hippocampus. So Papez's circuit is a network that connects the thalamus, mammillary bodies, and the hippocampus. Believed to be the key to forming long-term memories. This is very important right here. So we have the thalamus, mammillary bodies, and the hippocampus. Papez circuit, believed to be key to forming long-term memories. What about the prefrontal cortex? What about the prefrontal cortex, the PFC? Patients with prefrontal cortex damage have issues with the Wisconsin card sorting task. They have problems with the Wisconsin card sorting task. So patients with the prefrontal cortex damage have issues with the Wisconsin card sorting task. So short-term memory issues. Short-term memory issues. So prefrontal cortex damage has to do with the short-term memory issues. Remember, the Papez's circuit has to do with long-term forming memories. And that network there's a Papez circuit is connected to the thalamus, mammillary bodies, and the hippocampus. And yeah. So in order to form memories for short-term systems into long-term systems, you need a solid temporal, you need a solid hippocampus and a solid thalamus. Damage to those, you can't turn short-term memories into long-term memories. You found that out by monkeys that had damage to the hippocampus and the temporal lobe, and also damage to the thalamus. They cannot do this delayed non-matching to sample task, which required the ability to transfer memories from short-term systems into long-term systems. damage to the prefrontal cortex. You have issues with the Wisconsin card sorting task, just short-term memory issues. So what happened here? They with damage to this, they had damage with object permanence. Adult monkeys with prefrontal cortex damage don't seem to grasp object permanence. Prefrontal cortex is constantly developing throughout your youth and childhood. Object permanence usually develops in the first year of life. Think about it. For example, peekaboo with a baby. Let's say you're being funny with the baby, right? And then you cover your face. The baby thinks you're gone, or you're going like this with the baby, and then you hide where the baby can't see you, and you're just kneeling down in front of the bed. And the baby starts crying because they don't really understand that, one, humans can't disappear. We can't just with us. We don't disappear. We're not invisible. It's not magic. So that's lacking object permanence. As we get older, our brain develops and we have object permanence. If they do the peekaboo with us, we just laugh like, that's silly. You're not missing. But a baby would cry because they would think you disappeared, right? That's object permanence. So that's short-term memory. So damage to the prefrontal cortex. With the object permanence studies, adult monkeys with prefrontal cortex damage don't seem to grasp object permanence, right? So yeah, just think about that. So the basal ganglia. Lesions of the basal ganglia, like cuts to the basal ganglia impact procedural memories, but not explicit memories. So not sort of like remembering dates and facts and retaining memory that you read things, but procedural, like when you enumerate something, like first do this and then do that. Ride a bike, first get on it, put the helmet on. With procedural memories, so lesions to the basal ganglia impact procedural memories, but not explicit memories. The opposite of HM's problems. Diseases that lead to damage to the basal ganglia typically also have procedural memory deficits. Remember, basal ganglia has to do with Parkinson's disease. So diseases that lead to damage to the basal ganglia typically also have procedural memory deficits. Huntington's disease and Parkinson's disease. Need a new freaking highlighter. I have so many of them, but I seem to find them. Oh, there. So the cerebellum. We're focusing on the cerebellum now. Possibly key to procedural memories as well. So maybe also procedural memories. Motor learning. So the cerebellum is key, maybe, is also key to the procedural memories as well, but definitely motor learning. It is unclear and not currently agreed upon whether damage to the cerebellum leads to deficits in motor learning or simply the performance of that learned task. So it is unclear and not currently agreed upon whether damage to the cerebellum leads to deficits in motor learning or simply the performance of that learned task. Not sure what that means. So stress and memory. We know that stress impacts the amygdala, has connections to the hippocampus. Cortisol may damage the hippocampus. Repression, not sure repression exists. Flashbacks, flashbulb memories. So remember, stress and memory. We're not sure if repression exists or flashbacks or flashbulb memories. Cortisol may damage the hippocampus. All right, guys, we have this diagram. A little diagram right here. So learning and memory activity. Link the following types of memory to the appropriate brain area. We have procedural memory, short-term memory, motor learning, forming long-term memory. Then in the second table, list the evidence, task, patient that was studied that led to the conclusions. So brain area, the first one. Brain area, the hippocampus. Type of memory. So the hippocampus is responsible for forming long-term memory. The thalamus is also responsible for forming long-term memory. So hippocampus and thalamus forming the LTM. The basal ganglia is procedural memory. Remember, damage to that. Remember Parkinson's disease, which is a disease that affects the procedural movements. The mammillary bodies also forming long-term memory. Remember, the Papez's circuit was the connection between the thalamus, the mammillary bodies, and the hippocampus. That was for long-term memory. The prefrontal cortex is short-term memory. The cerebellum has to do with procedural. So they thought, right? Maybe even motor learning. And then also brain area. So what's the evidence? How do we know this? Because at the hippocampus, we know that it's forming long-term memory because of HM. Large areas of HM's hippocampi and temporal lobe, which is the hippocampus, were surgically removed. Loss of memories. So his short-term memory was okay. What does it say long-term memory, though, for the hippocampus? Well, more about his short-term memory was okay. Anyway, that was the right answer. So the thalamus, patient NA, and also DNMS task. Basal ganglia, we know that because we just went over it. That lesions of the basal ganglia impact procedural memories, but not explicit memories. And then we have basal ganglia evidence is Parkinson's and Huntington's disease. Mammillary bodies, Korsakoff syndrome, also affects the thalamus as well. So Korsakoff's, mammillary bodies. Remember, because of Papez's circuit, Korsakoff syndrome. Also, Korsakoff also deals with the thalamus as well. And then we have prefrontal cortex. That is the Wisconsin card sorting plus the object permanence, which is short-term memory. That's how we know. Anyone else want to look at it? 40 minutes. Learning and memory, guys. My first time going over that, looking it over after my lecture, and I'm definitely have to go over it again. Peace, y'all.